Welcome to Chapter 5, Microbial Metabolism. This chapter is about how microbes get energy, store it, and use it to grow, repair, and reproduce. Think of metabolism like a hospital's operations. It's everything happening behind the scenes that keeps the hospital running. If metabolism shuts down, the microbe or hospital can't function. Clinical relevance. Understanding metabolism is essential when considering how antibiotics work, how infections spread, and how we manage oxygenation, nutrition, and even fever in patients. Metabolism is the set of chemical reactions that keep a microbe alive. These reactions are tightly controlled, and their ultimate goal is reproduction, making more microbes. So if a microbe can metabolize well, it can grow and cause more trouble in the body. That's why we care. Our in practice tip. If you stop metabolism, you stop the microbe. That's what many antibiotics do, like stopping energy production in bacteria. NCLEX strategy. Always think, what disrupts the metabolism of a pathogen? If metabolism stops, so does the infection. Let's break down the 8 steps microbes follow for survival. They get nutrients, like we eat food. They need energy, which comes from light or breaking down food. They store that energy in ATP. Think of ATP like a charged phone battery. They break nutrients into smaller pieces called precursor metabolites. They use those pieces plus ATP plus enzymes to build bigger things. That's how they build macromolecules, proteins, DNA, cell walls. The microbe grows. Once big enough, it reproduces. This is like a baby growing into an adult, then having a baby of its own. But for bacteria, it can happen fast, in 20 minutes. Our in practice tip. This is how infections explode in the body. One becomes millions, especially in wounds, lungs, or catheters. This video on metabolism is available in the e-textbook. This video helps you visualize how metabolism works like a loop, taking in nutrients, creating energy, building cell parts, and reproducing. You'll see the process is like a factory assembly line, very efficient but also vulnerable to sabotage, like antibiotics or immune responses. Pharmacology insight. Sulfa drugs, for example, block enzymes needed for metabolism in bacteria, but not in human cells. That's selective toxicity. Metabolism has 2 sides, like yin and yang. Catabolism breaks big molecules into smaller ones. It releases energy, like burning firewood. Anabolism builds large molecules from small ones. It uses energy, like building a house. Bacteria use catabolism to make energy and anabolism to grow. Both are necessary, like a nurse needing both rest and energy. Nursing relevance. Fever increases catabolism. Burn patients have very high metabolic needs. They need more protein and calories to heal. This image shows how catabolism and anabolism are connected. The energy from catabolism powers the building process in anabolism. If one side fails, the whole system shuts down. NCLEX tip. Think of sepsis. In extreme cases, metabolism becomes dysregulated and energy is diverted from repair toward fighting infection. Oxidation, losing electrons. Reduction, gaining electrons. These reactions always happen together, like a seesaw. Microbes use redox reactions to move energy around, especially in cellular respiration. It's how they charge their ATP battery. Memorization tip, oil rig, which is oxidation is loss, reduction is gain of electrons. Clinical note? If oxygen is limited, hypoxia, microbial metabolism may shift, just like human cells do, leading to less energy and buildup of waste. Here you can see how electrons are transferred from a donor to an acceptor. This movement is where the energy comes from. Nursing connection. Some bacteria use alternate final electron acceptors, like nitrate or sulfate, especially in anaerobic environments like deep wounds or abscesses. Check out this video in your e-textbook, which shows how oxidation and reduction reactions are essential to metabolism. Just like in our own bodies, microbes use these reactions to survive and grow. Real-life microbiorelevance. M. tuberculosis thrives in oxygen-rich lungs for this very reason. It loves aerobic respiration. Microbes break down nutrients and store energy in the bonds of ATP. ATP is like a rechargeable battery, ready to power growth and reproduction. To make ATP, they use phosphorylation, adding a phosphate group to ADP. There are 3 ways this happens. 1. Substrate level phosphorylation, quick and direct. 2. Oxidative phosphorylation via electron transport. And 3. Photophosphorylation in light using microbes. Nursing tip. Think of energy-demanding processes in infection. Bacteria that grow fast need lots of ATP. Medications can target these steps. Now let's talk about enzymes, the tiny superheroes that make metabolism happen faster and more efficiently. Enzymes are organic catalysts. That means they speed up chemical reactions without being used up themselves. Just like nurses speed up healing by giving meds and care, enzymes help reactions go from hours to milliseconds. RN relevance. Certain antibiotics, like penicillin, target bacterial enzymes, disabling the microbe's ability to build or grow. Enzymes are often the targets in pharmacology. The video in your e-textbook explains how enzymes lower the activation energy. That's just a fancy way of saying they make it easier for reactions to start, like warming up before exercise. Without enzymes, microbial metabolism would be too slow to survive. Enzymes have different jobs depending on the type of chemical reaction they help. Hydrolases break things using water like digestive enzymes. Isomerases rearrange molecules. Ligases link things together. Lyases split molecules without water. Oxidoreductases handle redox reactions, energy production. Transferases move chemical groups from one molecule to another. You don't need to memorize them all, but know that different enzymes help with different tasks, just like the care team has RNs, RTs, and PTs. Test tip. Many antibiotics work by targeting specific enzymes in the bacteria's protein or cell wall synthesis process. This slide gives a chart that matches enzyme type to its function. For example, hydrolases are important for breaking down complex molecules. If these enzymes are blocked, the microbe can't digest nutrients or grow. Pharmacology application. Sulfa drugs interfere with an enzyme that helps bacteria make folic acid, essential for their DNA. Humans don't make folic acid, we eat it, so we aren't harmed. Enzymes are usually proteins. Some work alone, but others need help. The protein part by itself is called the apenzyme. It's like a nurse with no stethoscope, can't do much. Once it binds to a helper, like a cofactor, a vitamin, or metal ion, it becomes active. That complete active form is called a holoenzyme. Some enzymes are even made of RNA instead of protein, called ribozymes. Clinical connection. Many vitamins, like B vitamins, are cofactors. That's why patients with malnutrition or alcohol use disorder may have impaired metabolism and slower healing. This figure shows how a cofactor fits into the enzyme to activate it. Without the cofactor, the enzyme is useless. Like a locked toolbox, you need the key to get the job done. This table shows real examples like zinc, magnesium, or vitamins that help enzymes work. Some are minerals we absorb in food. Others come from supplements or the environment. Nursing assessment tip. Always assess nutrition status, especially in the elderly, alcoholics, and post-op patients. Enzyme function may be impaired if key vitamins, minerals are missing. This figure shows how enzymes make reactions easier. Think of it like pushing a wheelchair up a ramp instead of a staircase. The ramp enzyme lowers the energy needed. NCLEX insight. If a drug blocks the enzyme, the reaction doesn't happen. This is how antifungal or antibacterial drugs kill microbes. Enzymes are highly specific. Each one has an active site shaped to fit a specific molecule, like a lock and key. If the shape changes due to pH or temperature, the enzyme won't work. Fun fact, this is why fever or acidosis can affect enzyme function and make patients more prone to complications. Enzyme action has 4 key steps. Step 1, substrate binds to the enzyme. Step 2, enzyme changes shape slightly, induced fit. Step 3, reaction occurs, forming product. Step 4, released, an enzyme is ready again. This is like a nurse preparing a med, giving it, then resetting for the next patient. Real-life nursing analogy. Just like nurses don't work well without the right environment, sleep supplies, enzymes are also sensitive to their environment. This video in the e-textbook walks you through how enzymes do their job. They bind the substrate, do the chemical work, release the product, and reset. Enzymes are like efficient nurses on a shift. They care for one patient, then move to the next over and over. RN tip. Enzymes must be in the right condition to function. If the patient has a fever, acidosis, or electrolyte imbalance, these reactions can slow or stop even in human cells. Enzymes are picky. Their activity depends on temperature. Too hot, they denature, unfold. Too cold, they slow down. pH. Each enzyme has an ideal pH. If it's too acidic or basic, it won't work. Concentration. The more enzymes and substrates, the faster the reaction, up to a point. Inhibitors, chemicals that block or slow enzyme activity. Think of enzymes as Goldilocks. They need conditions to be just right. Clinical relevance. That's why fever, hypothermia, and acidosis affect not only human metabolism but also bacterial growth, and why nurses monitor these signs so closely. This figure shows enzyme activity peaking at the optimal temperature and pH, then dropping off sharply, like a bell curve. Enzyme activity is like an IV pump. Perfect flow rate helps, but too much or too little ruins the effect. NCLEX note. Always remember, enzymes can be slowed or deactivated by changes in the environment. A patient with sepsis, for example, may experience enzyme dysfunction due to acidosis or fever. Denaturation means the enzyme loses its shape. Since enzyme function depends on shape, like a key fitting a lock, denaturation shuts down the reaction. Heat, extreme pH, or toxins can cause this. Real-world nursing example. When cooking chicken to kill salmonella, you're using heat to denature bacterial enzymes, stopping their metabolism. These additional graphs drive the point home. There's an optimal range for everything. Too much of a good thing, like too high temp or too much acid, is still harmful. Fun fact for the NCLEX. Pepsin in the stomach works best at pH 2. But other enzymes, like those in the intestines, would fall apart in that environment. This shows how enzymes are tissue-specific. Some enzymes need a little push to turn on, like a nurse needs coffee. These pushes are activators which bind to a site on the enzyme, not the active site, and switch it on. This is called allosteric activation. Analogy: allosteric is like remote control activation. Not directly at the lock, but at a control panel nearby. This visual shows how the activator molecule binds to a different spot, changing the shape of the enzyme so it can now bind its substrate. Like reshaping a glove to better fit your hand. Pharmacology insight. Some drugs mimic these activators or block the enzyme at these remote sites. This is how we fine-tune enzyme function in both microbes and human cells. Inhibitors are substances that block enzyme function. There are two main types. Competitive inhibitors look like the real substrate and sit in the active site, blocking it. Non-competitive inhibitors bind somewhere else, allosteric site, and change the enzyme shape so it no longer works. This is how many antibiotics work. They block critical enzymes in bacteria. NCLEX connection. Always think, if I block this enzyme, what reaction fails? That's the MOA, mechanism of action, for many medications. Here we see a fake key, the competitive inhibitor, sitting in the enzyme's active site. The real substrate can't get in. This is like jamming the lock with gum. You can't unlock the door anymore. RN application? Sulfa drugs competitively inhibit an enzyme that bacteria need to make folic acid, a vitamin they need to grow and divide. Humans don't make folic acid. We get it from food, so we're safe. This animation shows competitive inhibition in motion. Picture a fake credit card swiping at a terminal. It blocks the real transaction. Pharmacology practice tip. Understanding enzyme inhibition helps you answer NCLEX pharmacology questions. Always look at what the drug is blocking and how that impacts the microbe or the human body. Now imagine a situation where the active site of the enzyme is wide open, but the enzyme still doesn't work. Why? Because something has bound to a different site on the enzyme, called an allosteric site, and changed its shape. This is non-competitive inhibition. The inhibitor isn't competing with the substrate, it's changing the lock entirely so the key no longer fits. Now imagine a situation where the active site of the enzyme is wide open, but the enzyme still doesn't work. Why? Because something has bound to a different site on the enzyme, called an allosteric site, and changed its shape. This is non-competitive inhibition. The inhibitor isn't competing with the substrate, it's changing the lock entirely so the key no longer fits. Analogy. It's like someone bending your car key remotely. The ignition is still there, but your key no longer works. RN practice. Some antiviral and antifungal drugs work by disabling critical microbial enzymes via non-competitive inhibition. This video in your e-textbook shows how non-competitive inhibition works. The inhibitor binds away from the active site, but it still disables the enzyme. You'll often see this in long-term feedback mechanisms, like when a cell wants to slow its metabolism down. Nursing tip. This is part of homeostasis. In both microbes and humans, sometimes cells need to slow down production. The body uses non-competitive inhibition for that. Here's where it gets clever. Microbes and our cells use something called feedback inhibition. When enough product has been made, that product feeds back and turns off the pathway, like a thermostat shutting off once the room is warm. The end product binds to an enzyme early in the chain, often using non-competitive inhibition to shut the process down. And Celek's tip. This is how cells avoid wasting energy. So if something disrupts this feedback, like in cancer cells or aggressive bacteria, metabolism becomes uncontrolled. Good question. Can oxidation happen without oxygen? Yes. Oxidation just means losing electrons, not necessarily using oxygen. Many bacteria use nitrate sulfate or even iron as the final electron acceptor. That's why some bacteria can thrive in deep wounds or inside abscesses where oxygen is limited. Nursing connection. This explains why anaerobic infections like Clostridium difficile or Bacteroides can still generate energy and cause major problems, even without oxygen. Let's talk about how microbes and human cells get energy from glucose, a sugar. Glucose is the most common fuel, and microbes break it down in one of two ways. Cellular respiration breaks it all the way down into CO2 plus water, lots of energy. Fermentation, partial breakdown, used when oxygen is low or absent, less energy. This is important because pathogens often switch between these modes depending on their environment. Clinical example? In deep wounds or diabetic ulcers, microbes may switch to fermentation, and that affects how fast they grow and how we treat them. This diagram gives you the big picture. Glucose is converted through a series of steps: glycolysis, Krebs cycle, and electron transport. If oxygen isn't available, it switches to fermentation. Key and Cleck's point. Microbes need to regenerate NAD+ to keep glycolysis running. Fermentation helps do that when there's no oxygen. Glycolysis happens in the cytoplasm of cells. It breaks one glucose molecule, 6-carbon, into 2 pyruvic acids, 3 carbons each. During this process, 2 ATP are invested, 4 ATP are produced, net gain equals 2 ATP. Also produces 2 NADH, electron carriers. This is fast and doesn't require oxygen, so it works for both aerobic and anaerobic microbes. Analogy. Think of glycolysis as splitting a $100 bill into smaller usable change, energy currency. This video in your e-textbook shows glycolysis in motion, breaking down glucose into pyruvate while charging up a few ATP batteries and NADH packs. It's like a mini power station. RN tip. Even when oxygen is low, like in a hypoxic wound, glycolysis can still provide energy. This is how anaerobes like Clostridium survive. Glycolysis has 3 main stages. 1. Energy investment stage. You spend 2 ATP to start the reaction, like paying to turn on the machine. 2. Lysis stage. The 6-carbon glucose is split in 2. 3. Energy conserving state. You gain 4 ATP and 2 NADH. Total profit. 2 ATP and 2 NADH per glucose. Clinical note? In fast-growing bacteria, glycolysis runs rapidly. If you inhibit it, the microbe can't survive. This is a target for antifungal and antimicrobial strategies. This figure shows the Embden-Meyerhoff-Parnas EMP pathway, fancy name for standard glycolysis. Remember, 1. It happens in the cytoplasm. 2. It works with or without oxygen. And 3. It is fast and essential for microbial survival. You'll also hear about alternate pathways like the Entner-Duderoff, but EMP is the most common. Takeaway? If you stop glycolysis, you stop the microbe. And some antibiotics do just that. This video in your e-textbook walks you through the 10 steps of glycolysis. You'll see how one glucose molecule is broken down into two pyruvate molecules, generating ATP and NADH. Focus on the big picture. Glycolysis is fast, doesn't need oxygen, and is the starting point for both respiration and fermentation. Tip: don't get lost in every enzyme. Just know the flow. Glucose becomes pyruvate plus a little ATP plus NADH. This diagram shows substrate-level phosphorylation, which is how ATP is made directly in glycolysis. Imagine you're handing a phosphate group from one molecule directly to ADP to make ATP, no oxygen or mitochondria needed. This is different from oxidative phosphorylation, which requires the electron transport chain. Analogy: like charging your phone directly from a power bank, substrate level, not from a wall outlet, oxidative phosphorylation. Or an insight: in anaerobic infections or during intense exercise, this is how cells and microbes keep making small amounts of energy without oxygen. In your e-textbook, you can find this case study. This case is about Jane's father who has tuberculosis. He was previously treated, but now it's back. Why? Mycobacterium tuberculosis can become dormant, hiding in lung granulomas called tubercles. When oxygen is low, they shut down and wait. When conditions change, like a weakened immune system, they wake up and use aerobic respiration to multiply again. Nursing relevance. This is why latent TB can become active TB later in life, especially in older adults or immunocompromised patients. NCLEX tip: always assess for reactivation of TB in patients with immune suppression, HIV, chemo, elderly. If oxygen is available, microbes go beyond glycolysis into cellular respiration, which is way more efficient. It includes 1, synthesis of acetyl-CoA, 2, Krebs cycle, and 3, electron transport chain. These steps happen in a specific order, generating a lot more ATP. Fun fact: aerobic respiration yields about 34 ATP, while fermentation yields only 2. This enzyme complex connects glycolysis to the Krebs cycle by converting pyruvate into acetyl-CoA, producing CO2 and NADH in the process. It's like prepping ingredients for the cooking stage, Krebs cycle. It happens right after glycolysis. Think of it as step 1.5. Clinical relevance. Many toxins and antibiotics block this step, cutting off energy supply and killing the microbe. Each glucose yields 2 pyruvates, which gives 2 acetyl-CoA, 2 CO2, and 2 NADH. This is the prep step before the Krebs cycle. The NADH produced will later be used to make more ATP in the electron transport chain. NCLEX connection. Disruption here, via drugs or toxins, can crash the whole energy system of the microbe. The Krebs cycle, aka citric acid cycle, is where most of the energy-rich molecules, NADH, FADH2, are made. These will fuel the electron transport chain. It happens in the cytosol for prokaryotes and the mitochondrial matrix in eukaryotes. Microbes rely on this step to generate electron carriers. It's not about making a lot of ATP here, but about making fuel for the next step. Analogy: Krebs is like a fuel refinery, producing fuel NADH, FADH2, not electricity, ATP, directly. The Krebs cycle includes 6 types of reactions. 1, anabolism building. 2, isomerization reshuffling atoms. 3, redox transferring electrons. 4, decarboxylation releasing CO2. 5, substrate level phosphorylation making a bit of ATP. And 6, hydration adding water. It's a chemical dance that repeats with each turn of the cycle. Clinical connection. Some chemotherapy agents block enzymes in this cycle to stop rapidly dividing cancer cells or microbes. This figure shows the full Krebs cycle. Focus on what comes out of each turn, from 1 acetyl-CoA, 3 NADH, 1 FADH2, and 1 ATP, 2 CO2. Since 1 glucose gives 2 acetyl-CoA, the cycle turns twice per glucose. Test strategy. Always double numbers for total yield per glucose. This is a common NCLEX trap. This video in your e-textbook walks through the Krebs cycle in motion. Remember, this is where electron carriers are loaded up with energy, ready to drop off at the electron transport chain. The big payoff comes later, but this step is essential. Takeaway: if you block the Krebs cycle, you stop the microbe from charging its batteries. That's a powerful target for medications. NCLEX Rand-style practice question. Question. A nursing student asks why aerobic bacteria produce more energy than anaerobic bacteria. What is the best response? A. Anaerobic bacteria do not undergo glycolysis. B. Aerobic respiration includes the Krebs cycle and electron transport chain. C. Anaerobic bacteria produce more ATP through fermentation. D. Oxygen directly produces ATP in aerobic bacteria. Correct answer B. Aerobic respiration includes the Krebs cycle and electron transport chain. Rationale. Aerobic bacteria generate significantly more ATP because they complete all steps of respiration, glycolysis, Krebs cycle, and electron transport. Anaerobic bacteria typically rely on fermentation or alternate acceptors, producing far less ATP. Another video in your e-textbook shows each reaction in the Krebs cycle. Even if you don't memorize every enzyme, remember? 1. The cycle takes acetyl-CoA and extracts high-energy electrons in NADH and FADH2. 2. These electrons are fuel for the next step, the electron transport chain. Think of the Krebs cycle as the prep kitchen. It doesn't serve the final dish, ATP, but it preps all the ingredients for it. Key NCLEX point, the Krebs cycle depends on oxygen indirectly. Without oxygen at the end of the ETC, the cycle backs up and stops. For every glucose, 2 turns of the cycle, we get 2 ATP, 6 NADH, 2 FADH2, and 4 CO2, waste gas we breathe out. The ATP is nice, but the NADH and FADH2 are the real jackpot. They'll make a huge ATP payout in the ETC. RN connection. If oxygen delivery to tissues is impaired, shock, hypoxia, NADH can't unload its electrons. The cycle slows and ATP production drops, leading to organ failure. The electron transport chain, ETC, is where the magic happens. Most ATP is made here. NADH and FADH2 donate their electrons to a chain of proteins. These electrons move down the chain, releasing energy used to pump protons, H+, across the membrane. This creates a proton gradient, like water behind a dam. The cell then uses this stored energy to make ATP. Analogy, the ETC is like a hydroelectric power plant. The electrons are the water flowing downhill, and the turbines are ATP synthase. This diagram shows one possible layout of the ETC in bacteria. In prokaryotes, the ETC is in the cytoplasmic membrane. In eukaryotes, it's in the inner mitochondrial membrane. Knowing where this happens matters because antibiotics can target bacterial ETC proteins without harming human ones. In this animation in the e-textbook, watch how electrons move from NADH/FADH2 down the chain, releasing energy in steps. This controlled release prevents wasting energy as heat and maximizes ATP production. Clinical link. Cyanide poisoning stops the ETC cold by blocking the final step. Oxygen can't accept electrons and ATP production stops. This is why it's fatal in minutes. In aerobic respiration, oxygen is the final electron acceptor. It becomes water after picking up electrons and protons. In anaerobic respiration, microbes use something else, like nitrate, sulfate, or even carbon dioxide. That's how they survive in oxygen-free environments. NCLEX relevance. This explains why certain infections thrive in deep wounds or abscesses where oxygen delivery is poor. Here's another possible ETC layout. Microbes can have different chain components depending on their environment. This flexibility makes some pathogens harder to kill because they can switch energy strategies. This video in your e-textbook shows electrons moving step by step, pumping protons into the periplasmic space or mitochondrial intermembrane space. This stored proton gradient is the key to ATP synthesis. Nursing insight. Mitochondrial diseases in humans and some antibiotic side effects are related to ETC dysfunction, leading to fatigue and muscle weakness. ATP yield can vary because 1. Not all protons are used to make ATP. Some power other cell functions. 2. Some NADH is spent moving things across membranes. And 3. Damage to membranes, like fever-induced denaturation or antibiotic effects, can leak protons, lowering yield. NCLEX tip. Fever can speed metabolism but also damage enzymes and membranes, lowering efficiency. Chemiosmosis is the process of using the proton gradient to drive ATP production. Protons flow back down their gradient through ATP synthase, a protein turbine. This spins and cranks out ATP from ADP and phosphate. This is oxidative phosphorylation because the gradient was created by oxidation in the ETC. One glucose molecule in bacteria can yield approximately 34 ATP from this step alone. RN connection. In septic shock, membrane integrity and oxygen use are disrupted. Chemiosmosis fails and ATP levels plummet, leading to multi-organ failure. This table summarizes what we've been building up to, the total ATP yield from one glucose in a bacterium. Glycolysis, 4 ATP substrate level and 2 NADH. Krebs cycle, 2 ATP substrate level, 8 NADH and 2 FADH2. ETC, 34 ATP. Total about 38 ATP in prokaryotes. Humans usually make about 36 ATP per glucose because moving NADH into mitochondria costs some energy. NCLEX connection. In oxygen-deficient states, we lose the ETC step, so instead of 38 ATP, we only make 2 ATP via glycolysis. That's why hypoxia quickly becomes life-threatening. Not all microbes use the standard glycolysis pathway. Some use Entner-Doudoroff ED pathway, which occurs only in some bacteria, and it yields 1 ATP, 1 NADH, 1 NADPH. Some use pentose phosphate pathway, which produces building blocks for DNA, RNA, and certain fats. Not much ATP. Why does this matter? Because these alternate pathways can make pathogens harder to target. They don't rely entirely on glycolysis. Example, Pseudomonas uses ED pathway, part of why it can survive in harsh environments and resist some drugs. When microbes can't use the Krebs cycle or ETC, like in oxygen-poor wounds, they switch to fermentation. In fermentation, glycolysis makes 2 ATP. NADH dumps electrons onto organic molecules like pyruvate to regenerate NAD+. Fermentation produces acids, alcohols, or gases as byproducts. This keeps glycolysis going even without oxygen. Clinical note, infections with foul odors often involve anaerobic fermentation. Clostridium species produce smelly butyric acid. This figure shows some examples of fermentation. Lactic acid fermentation, like in muscle fatigue or lactobacillus. Alcohol fermentation, Saccharomyces. And mixed acid fermentation, E. coli. These end products can be used in lab tests to identify microbes. NCLEX tip, lactic acid buildup in humans, like in shock, is a warning sign of anaerobic metabolism due to poor oxygen delivery. Let's compare. In aerobic respiration, O2 is the final acceptor and it has high ATP yield. Anaerobic respiration, other molecule as final acceptor with moderate ATP yield. Fermentation, organic molecule as final acceptor, low ATP yield. For nurses, this explains why certain infections, anaerobes, grow slowly. They make less energy. Fermentation doesn't just keep microbes alive without oxygen. It's behind a lot of foods and everyday chemicals you know. For example, Swiss cheese, Propionibacterium, now often labeled Cutibacterium, ferments lactate into propionic acid and CO2. The CO2 makes the eyes, holes. Propionic acid gives the nutty flavor. Yogurt and many cheeses, soy sauce. Lactobacillus or Streptococcus make lactic acid, dropping pH to thicken milk proteins and create tangy flavor. Wine, beer, and bread. Saccharomyces cerevisiae produces ethanol plus CO2. CO2 leavens bread. Ethanol is the alcohol in wine, beer. Industrial solvents, Clostridium, for example, C. acetobutylicum, perform ABE fermentation, generating acetone, think nail polish remover, and isopropanol, rubbing alcohol, along with butanol. This video in your e-textbook reinforces the process. First glycolysis, then NADH passes electrons to pyruvate, next regenerates NAD+, so ATP production continues without oxygen. The downside? Very little ATP, but enough to survive. This is a good question to think about. Why do electrons carried by NADH allow for production of 50% more ATP molecules than do electrons carried by FADH2? Well, NADH donates electrons earlier in the ETC, pumping more protons and making more ATP, about 3 per NADH, compared to FADH2, about 2 ATP. This explains why starting point in the ETC affects yield. Test tip. If a drug blocks NADH dehydrogenase, first step of ETC, ATP yield drops significantly, affecting both human and bacterial cells. Microbes don't just eat sugar, they can catabolize lipids and proteins too. Lipids can be broken down into glycerol, goes into glycolysis, and fatty acids, broken into acetyl-CoA by B-oxidation. Proteins can be broken into amino acids, deaminated to remove nitrogen, then fed into Krebs cycle. This flexibility helps pathogens survive when glucose is scarce. RN connection. In prolonged illness or starvation, humans switch to burning fat and protein, causing weight loss and muscle wasting. This figure shows how fats yield huge energy. Each fatty acid chain can generate many acetyl-CoA molecules, producing lots of ATP. That's why fats are high calorie and why lipid-rich bacteria like Mycobacterium tuberculosis can store energy for long dormancy. Here we see how proteins are broken down. Proteins are first digested into amino acids by proteases. Amino acids are deaminated, nitrogen group removed, producing ammonia and organic acids. The organic acids feed into the Krebs cycle. For microbes, this means they can grow even when sugar and fat are scarce. RN relevance? In humans, excess protein breakdown releases nitrogen waste as urea in the liver or ammonia in microbes. This can cause ammonia buildup in infections and in liver failure. Why does protein catabolism produce ammonia? Because the amino group, NH2, from amino acids is converted into ammonia during deamination. In humans, ammonia is toxic and must be converted to urea by the liver. In some bacteria, ammonia is simply released into their environment. NCLEX tip. Patients with liver failure can develop hepatic encephalopathy because ammonia builds up in the blood, affecting brain function. Many organisms, including some bacteria, can make their own organic molecules from CO2 by capturing light energy. This is called photosynthesis. It's not just for plants, cyanobacteria and algae do it too. They use light to power the production of glucose, which can later be broken down for ATP. Fun fact, some photosynthetic bacteria are found in freshwater, salt water, and even hot springs. This video in your e-textbook walks through photosynthesis. Light-dependent reactions capture light to make ATP and NADPH. Light-independent reactions, Calvin cycle, use that energy to make glucose from CO2. In bacteria, this may happen in folded parts of their cell membrane instead of chloroplasts. This video in your e-textbook connects the dots between microbial metabolism and photosynthesis, showing how energy can be captured in many ways, whether from light, sugars, or other chemicals. Photosynthetic microbes have pigments called chlorophylls that absorb light energy. They have a light-absorbing head with magnesium at the center. Different chlorophylls absorb different wavelengths, letting microbes live in different light environments. They work much like cytochromes in the ETC. NCLEX connection. Magnesium is essential for chlorophyll function. Malnutrition in plants leads to yellowing leaves. In human nutrition, magnesium is also critical for enzymes and muscle contraction. Pigments are arranged in photosystems, antenna-like structures that capture light. In bacteria, these are embedded in the cell membrane. In eukaryotes, they're inside chloroplasts. Two main types, photosystem 1 PSI and photosystem 2 PS2. Both are needed for maximum energy capture. Analogy? Think of them like satellite dishes picking up light signals. This figure shows how bacterial membranes fold inward to increase surface area for photosynthesis, kind of like the villi in your intestines increase absorption surface. Photosystems use light energy to power redox reactions, producing ATP and NADPH. This is the light-dependent phase, where sunlight is converted into a form the cell can store and use. Clinical connection. This process is a model for photodynamic therapy in medicine, where light activates drugs to kill cancer cells or microbes. Two kinds of light reactions. Cyclic photophosphorylation, electrons cycle back to PS1, ATP only. And non-cyclic photophosphorylation, electrons flow from water through PS2 and PS1, producing ATP, NADPH, and oxygen. Bacteria without PS2 can't split water or produce oxygen. These are called anoxygenic phototrophs. Here's a close-up of the reaction center, the heart of a photosystem. Light energy hits pigments. Electrons get excited and jump to a higher energy state. These electrons move down in ETC, like in respiration, to make ATP or NADPH. The idea is the same. Move electrons, pump protons, make ATP. There are two versions. 1. Cyclic photophosphorylation uses only PS1. Electrons cycle back to the same photosystem and it produces ATP only, no NADPH, no oxygen. 2. Non-cyclic photophosphorylation. It uses both PS2 and PS1. Electrons come from water, producing ATP, NADPH, and oxygen. Oxygen comes from splitting water in PS2. Test tip. Remember, non-cyclic is the only one that produces oxygen. This animation in your e-textbook shows cyclic flow. Think of it as running on a treadmill. You get energy, ATP, but you end up back where you started. Here's non-cyclic, more like a road trip. Electrons start in water, travel through PS2 and PS1, and end up in NADPH. Along the way, ATP and oxygen are produced. This mirrors how our mitochondria operate. Electrons flow in one direction and end up in a final electron acceptor. Quick review. Substrate level, direct phosphate transfer, glycolysis, Krebs, oxidative, ETC, and respiration. Photophosphorylation, ETC, and photosynthesis using light. For the NCLEX, know the definitions and which processes they occur in. Light independent means they don't need light directly. They use ATP and NADPH from light-dependent reactions. This is where carbon dioxide gets fixed into glucose. There are 3 stages. Stage 1, fixation, CO2 joins with RUBP. Stage 2, reduction, energy from ATP and NADPH turns it into sugar. Stage 3, regeneration, RUBP is remade so the cycle can continue. Analogy? It's like a bakery that uses the electricity stored earlier, ATP, to bake bread, glucose, all day. This simplified diagram shows CO2 coming in and glucose coming out, powered by ATP and NADPH. Remember, this is an anabolic pathway building large molecules from smaller ones. This animation walks through the Calvin cycle steps. For nurses, the important part is knowing that ATP and NADPH are the universal energy currency, whether made in photosynthesis or respiration. This is a good question. Why is the student incorrect? It's not simply respiration backwards. Respiration breaks down glucose to release energy. The Calvin cycle uses energy to build glucose. Different enzymes and pathways are involved. NCLEX tip. Anabolic is not reverse catabolic. They are separate processes that may share intermediates but not identical steps. Anabolism means building. These reactions require energy from ATP and precursor metabolites. Some are reverse versions of catabolic steps, but many are unique. Amphibolic pathways can run both directions depending on the cell's needs. For microbes, anabolic capacity determines how fast they can repair and grow. RN relevance? Wound healing in humans is an anabolic process requiring good nutrition and energy. Malnourished patients heal slower because anabolic pathways are impaired. These 12 molecules are the building blocks for everything a cell needs: amino acids, nucleotides, lipids, and more. They're made from pathways like glycolysis, the Krebs cycle, and the pentose phosphate pathway. Without these precursors, a cell can't grow or reproduce. RN relevance? In malnutrition or chronic illness, the body can't produce enough of these precursors for wound healing, especially proteins and nucleotides for tissue repair. Gluconeogenesis is making glucose from non-carbohydrates like amino acids or glycerol. This is anabolic and requires ATP. It's important in fasting and for some microbes that need glucose but don't have a sugar source. NCLEX tip. Steroids increase gluconeogenesis in humans, which is why long-term steroid use can cause high blood sugar. This shows how microbes and us build triglycerides from glycerol and fatty acids. Glycerol often comes from glycolysis intermediates. Fatty acids come from acetyl-CoA. Lipids store energy and form membranes, which is essential for survival. Clinical link. Malnutrition can impair lipid production, leading to weak cell membranes and poor immune function. Amino acids can be made by amination, adding an amino group, or transamination, transferring it from one molecule to another. Nitrogen is required, so if it's missing, protein synthesis slows or stops. NCLEX connection. This is why nitrogen balance matters in burn patients and post-surgical recovery. They need lots of amino acids for tissue repair. Clinical link. Malnutrition can impair lipid production, leading to weak cell membranes and poor immune function. Nucleotides, building blocks of DNA, RNA, come from glycolysis and Krebs cycle intermediates plus nitrogen bases. If nucleotide synthesis is blocked, cells can't divide. Pharmacology tip: some antiviral and cancer drugs block nucleotide biosynthesis, stopping replication of viruses or rapidly dividing cells. Here's another question to think about. Why is nitrogen required for the production of amino acids by amination? Nitrogen is essential for amino acids and nucleotides. Without nitrogen, no proteins or DNA can be made. That's why nitrogen deficiency slows growth in both microbes and humans. Clinical note: in protein energy malnutrition, kwashiorkor, nitrogen deficiency is severe, leading to edema, poor immunity, and delayed wound healing. Cells are smart. They use the most efficient energy source first. They only make enzymes when their substrate is available. And they stop making products when enough has been produced. In eukaryotic cells, enzymes for different pathways are kept in separate compartments, organelles. This prevents interference and allows fine control. N-selects tie-in. This separation explains why certain poisons or antibiotics are selective. They may target bacterial cytoplasm, but not human mitochondria. Enzymes can be controlled instantly. Allosteric activation inhibition changes enzyme shape, and feedback inhibition shuts down a pathway when enough product is made. Pharmacology insight: statins, cholesterol-lowering drugs, work by feedback inhibition, blocking the first enzyme in cholesterol synthesis. This big picture diagram shows how all major pathways, glycolysis, Krebs, photosynthesis, fermentation, are interconnected. In a microbe, these pathways can shift depending on oxygen, nutrients, or stress. NCLEX strategy: when thinking about infection treatment, ask, if I block one pathway, does the organism have an alternate route? This video in your e-textbook ties it all together. Metabolism is not one straight line. It's a network. Microbes can reroute energy flow to survive stress. This adaptability is why infections can be hard to eradicate. This is the last tell my why question of this chapter. Why is feedback inhibition necessary for controlling anabolic pathways? That is because feedback inhibition prevents waste. If a cell kept producing a molecule it didn't need, it would waste energy and resources, lowering survival chances. In medicine, understanding feedback is key to knowing how drugs and hormones regulate metabolism. NCLEX Ren style practice question. Question: a nurse is caring for a patient